So what are bioreactor grown mitochondria? And more importantly, what is mitochondrial transplantation? And by the way, this is, of course, on its way to human tests, but not there. So don't go out and try to inject mitochondria. I just want to say that we have a tremendous team, and I put the company together 3 years ago by basically going around to all these brilliant people, mostly in North America, who had been already working on mitochondrial transplantation. This is something that's been around for a few years now. And the analogy that we like to use is, this is my actual car. It's an incredibly ugly 25-year-old Toyota minivan, that won't die. And I think everybody has one of these. And of course, the kids, we used it over the years to carry the kids everywhere. But the key thing with this car is that it would never stop running. It's still out there and it still runs. But the key is you have to replace the battery every 4 years. That's what we learned. The battery would die. And why? Because car batteries, the electrodes corrode and the chemicals go bad. And used to be you could refill them with new chemicals. Now they just don't bother. They just pull it out and put a new one in. And so the analogy here is that when we're talking about mitochondria, mitochondria, they're called the powerhouses of the cell. Sometimes I call them the replaceable batteries of the cell, like those little batteries you snap into your drill. It turns out that the human body can have those mitochondria replaced, which is not anywhere in any textbooks yet. It's this what's come out of the last 10 years of mitochondrial research. So what are mitochondria? They are tiny little organelles inside each of our cells. They do all the energy generation for the cell. So you typically have about 100 mitochondria in your cell. They kind of move around, and they combust the glucose and the oxygen, and they make ATP, and that's what the cell uses to do everything it does. And they have their own tiny little mitochondrial DNA, which are rings. And the nuclear DNA is 3 billion base pairs. The mitochondrial DNA is only 16,000 base pairs. So that's a good example of just how small they are. And they're very similar in a lot of ways to what you see in bacteria, because that's originally where mitochondria came from. Originally, the theory is that they were bacteria floating around a billion years ago, and they joined in with the DNA and made us. A billion years later of mitochondrial and nuclear DNA evolution has produced us, right? So what doctors know this, and this has been studied for many years, mitochondrial decline as we age is one of the underlying reasons for many, many, many diseases. And it's really very simple. Your cells run on energy. And when you're 20, of course, that's at your peak. That's when you're full size, your mitochondria are in perfect condition. The mitochondrial DNA are all in perfect condition, but that declines as we get older. And we're learning a lot about how that decline works. And it causes all these diseases because there's just not enough energy for the cells to do their job properly. Okay. It's really just that simple. You might have other reasons for getting diseases. You might be predisposed towards cardiovascular disease or Alzheimer's or any of these other things, diabetes, arthritis. But the mitochondrial energy decline is what kind of triggers it. So why do mitochondria decline? And this is something that we've actually been studying as part of our work. I just want to show you a mitochondrial DNA scan. And this actually is my mother, okay, who is now 94, who was kind enough to let me analyze her mitochondria. And what you're seeing here is actually the DNA structure of her mitochondria we got from a urine sample. Each gray line, you can kind of see these gray lines across. Each of those is one strand of mitochondrial DNA. We got about 500 of them, and we sequence them and we lay them in a plot. This is a very common way of doing it. And what you see here in this plot, of course, the genes, mitochondrial DNA encodes about 10 or 12 genes. But what you see are all these little dots and lines. These are all errors. Okay, these are what we call damaged sectors. It's like sectors on a hard disk. As you get older, your mitochondrial DNA is replicating constantly, and every replication, there's a chance of a little bit of an error creeping in. So my mom actually has a damage score of 25%. And then this is mine. And by the way, mitochondria are passed down from the mother to the child with absolutely no modification. There's about 500,000 mitochondrial DNA in each egg cell. The nuclear DNA comes from the father and the mother. The mitochondrial DNA just comes from the mother. And so I have exactly the same as my mother. So in theory, it should look the same. But you can see the difference. I only have an 11% damage score. Okay. This is my nephew, who also has exactly the same mitochondria because he's descended through my sister. He has a 7% damage score. And so what we can do is we can look at the damage in the mitochondrial DNA, and we actually can see with a lot of massaging, we were able to get a line here that shows the decline over time. And it's actually at the point where you can almost predict at what point someone, if they manage to dodge all the other diseases of aging, there's a certain point when you hit the point where your mitochondria just can't do the job anymore. And that's when people they just fade away, and that's when they pass away of old age. Okay. So, of course, there's a lot of other pieces of aging. There's a lot of other things in aging, but the mitochondrial component is its own aging curve, which is very, very important. The other thing is that can accelerate. If you drink a lot, if you smoke, if you have other kinds of diseases, like people who have multiple sclerosis, for example, if you have a disease for 30 years that you're fighting, a chronic disease, then your body's constantly battling. And what happens is your mitochondria get burned out faster. And so you age quicker. And that's why you see people will die 20 or 30 years younger. Of course, there's many other reasons for that. You might get cancer, or you might get an injury, but that's, again, this question of how long are your mitochondria going to last you if you were starting with perfect ones when you're young. So what's the purpose of a mitochondrial bioreactor? Basically, our idea is, hey, let's go in every 10 years or every 5 years or whatever it turns out to be, and let's give you a boost of new mitochondria. If by 75 years old, we think you've lost as many as 50% of your healthy mitochondria. Either they've been disposed of or they've gotten damaged. If we give you a boost of another 20% of young, healthy mitochondria that we've grown externally, then in theory, we could prevent you from developing these aging diseases or even reverse them, okay? And you can see the chart goes out here into the hundreds. That's my hopeful side says, gee, maybe this will give us longer lifespans, but we're not counting on that. But, we're focused on the healing component primarily, but lifespan is a possibility. So, of course, everybody says, well, wait a minute, how do you transplant mitochondria, right? We can transplant livers, we can transplant hearts. How do we transplant mitochondria? And this is something that has come out in the last 8 years or so. And it turns out that it is actually quite doable. The way it would look to you is it would look like a blood transfusion, okay? And of course, in the behind the scenes, it's much more complicated than that. It turns out that mitochondrial transplantation, mitochondrial transfer is happening in your body constantly. As you sit here right now, there are billions and billions of mitochondria being shifted around via your bloodstream and also in your brain and in your cardiac tissue. Mitochondrial transplantation is, as we say, pervasive and evolutionarily conserved. It happens everywhere in the body. And this is completely new science. You're not going to see any of this in the traditional high school textbooks. Most people think that mitochondria just sit there in the cell. They're constantly in motion. And we've proven this over and over again. And there's actually people doing this in human patients now. It's being used to treat diseases. It's being used to treat people, for instance, who are getting heart surgery. So the technique that we're using, that we're leveraging to do our technology is something we call mitelets. It turns out that if your blood is full of platelets, which are usually used to create clots, and the platelets are created by the bone marrow, you create 100 million platelets every day, and then 100 million are disposed of after about 7 to 10 days. They get thrown away. They're the most numerous blood cell in your blood, and they contain a whole bunch of mitochondria, 5 per platelet. And so we found that after those platelets are activated, they spit those mitochondria out in little vesicles, which we call mitelets. And those vesicles are absorbed by other white blood cells or platelets. So they're like little battery packs that the white blood cells and the platelets can exchange, and they're being recycled because mitochondria are extremely valuable, and you don't want to lose any of them. Okay, the body is evolved to preserve mitochondria whenever possible because they're very valuable, and your survival as a species, if you're running away to escape a leopard, your mitochondria are what get you the energy to escape. So they are pure survival, and your body conserves them all the time. Here's an example of some testing we did where we showed, or another team did, where they found that these mitelets decline as you get older, right? And so we think that the decline of these mitochondria and the decline of the quality is a fundamental cause of aging. You just don't have as many of them supplying new mitochondria to your cells. And those mitochondria in your cells tend to burn out. They burn out after 10 or 15 years. The mitochondrial DNA aren't any good anymore. And so your body is supplying new ones to supplement them. So if we're going to cure diseases, how do we do that? There is no supplies of mitochondria. I mean, we're not going to go out and squeeze mitochondria out of teenagers. That wouldn't be popular at all. So we have to figure out some way to grow them. And that's fundamental to any kind of adult disease treatment. And so we are developing bioreactors, which are basically stem cell bioreactors that are specialized for growing and harvesting mitochondria. We have a technology for encasing them in a specialized coating, a protein coating, so that they're protected, and then you just infuse them. That protein coating has receptors in it, so we can target the mitochondria to go to specific organs. Okay, so they could go to your heart, your liver, your whole body. It's whatever we choose. And of course, you can also inject them in different places. So I'll show you some of those tests. So if you were going to get a bioreactor treatment, let's say, 5-10 years from now, it would be very much similar to a lot of the kind of genetic treatments that they're already doing. You go in, you give some blood, they take your mitochondria from your blood, we grow them in massive quantities, we coat them, and then we put them back into your body. Okay? Alternatively, we might mass produce them with some sort of common format. Turns out there's not very many types of mitochondria. There's 26 major groups that kind of spread across the world, depending on the migration of the human species. Different ethnic groups have different mitochondrial types. So it may be that we could create a storehouse of a couple dozen variations, and we could use those instead of creating them from scratch. So we've been around for 3 years. We've been doing a whole bunch of testing of this concept. A lot of other people in other universities are doing similar things. Here's one that I like. We took mice at University of Kentucky. We injected them with sepsis. So we basically gave them sepsis, which would normally be fatal in 24 hours. That's a bacterial infection. These were older mice, 13 months old. And then we gave them massive injections of these mitelets that we collected from the platelet donations that we got from young mice. So we got hundreds of these very young mice and got platelet donations from them. And extracted the mitelets and we injected them into these older mice. And when they get these mitelet injections, they survive. That's the blue line here. Okay. So it's this amazing thing that you could effectively, we're giving them the same immune system that they had when they were younger. We're reversing the age of the immune system. People say, well, is that really reversing the age? I don't know. It's hard to say. What is age, right? Is age just energy? In some cases, maybe it is. I'll be talking more about that later at the end of the session. I'll talk about longevity. We also did a thing where we just injected mice with a whole bunch of these mitelets to see what would happen, and we got they got stronger. They started running on their wheels like crazy. They grow back their brain tissue, and that's something that Ben will be talking about in about half an hour. So it's really a way of making them basically become younger again. And by the way, we've also extended this work now, just started in dogs. We did a test where we injected a bunch of mitochondria into the eye, into the retina of some mice. This red stripe down here is what's called the RPE layer, and those are red fluorescent mitochondria. And this blue tissue is the retina. And you can see we injected them in one part of the retina and they spread all the way across. They actually end up going down into the optic nerve. They'll go up into the cornea. Mitochondria just kind of spread in the whole eyeball. Basically, the goal is to make the eyeball younger. Okay. So anyway, that's our work. That's what we're working on. And the goal here is to get this into the immune system tests, retina, skin, muscle. There's a whole bunch of different what we call indications there that can be tested on and could be potentially cured. Okay. I always tell people to remember this is already in use in human beings. People always say, oh, how long till it's in human beings? There are doctors using mitochondrial transplants right now in heart surgery. They have used them in some rare childhood diseases. It's very, very new. It's very rare. We're trying to move it into the bigger world by growing them in bioreactors. And obviously, we would like to have a worldwide network of these bioreactors providing supplemental mitochondria for everybody. And so in about half an hour or an hour from now, I'm going to talk more about how that could apply to longevity. And by the way, I just mentioned, yes, we are raising funds. So there's my email address if anybody wants to contact me.
Tom, you've got 4 minutes. Maybe we can talk about longevity now, so we can just go straight into the questions at the end.
Sure, absolutely. Well, let me just go back and talk about that picture there. I actually have a pretty, I have 10 or 15 minutes of longevity stuff, so I'll just give you a preview of that. So the way that we look at longevity is, and people say, oh, there's health span and there's lifespan. And I don't make a big differential between them. I think that if we want to improve people's health at 70 years old, this is the way to do it, because everybody knows you hit about 70, 75, 80 years old, and it doesn't matter what you've done, doesn't matter how hard you work, you just start to lose ground. And then by the time you hit 90, boom, you're really losing ground fast. I mean, my dad was skiing when he was 90, and then he finally had to quit. He said, I just don't have the leg muscles anymore. Your leg muscles actually decline 1% a year after you're 30 years old. So we think that mitochondrial transplants are going to be absolutely critical for any kind of longevity treatment, whether it's for 80-year-olds or for giving people longer lifespan. Because if you have 50% of the mitochondria you need in your body at the age of 80, you're not going to be able to regain any ground. If we want to reverse the aging process or even slow it down, we've got to restore those mitochondria. And I always tell people, again, it's like thinking of if you think about refurbishing a laptop, here's another battery analogy. You always replace the battery in a laptop when you rebuild it. So anybody who's ever bought one of those refurbished laptops from online, They always put a new battery in, because if you don't have power, you can't do anything else. Yes, we're going to clean it. We're going to clean the keyboard. We're going to install a new operating system. Maybe we'll put more RAM in there, but you're always going to put in a new battery. That's the starting point. And so that's my argument for longevity, is that mitochondrial transplant will be a foundational component of all longevity treatments, and also a foundational component of a whole bunch of just plain medical treatments. We think you'll be hearing a lot more about this. I think that there's an enormous number of medical conditions that are unsolvable and incurable because we haven't addressed the loss of energy. And if we address that, then we will get a handle on them. And people are talking about Alzheimer's. Alzheimer's is a very big target for mitochondrial work. We're talking about eye disease, AMD, glaucoma. Those are all big targets for mitochondria.
Well, it's a delight to be with you, and great to see you again, Ranjan. My introduction to this team is through the Global Health Span Summit that was held in Riyadh in December of last year, and so it's my privilege to be here with such a distinguished group of folks. We're going to take quite a detour and talk about space flight, actually. So let me pull this up. Here we go. Stand by one.
And Tom, you can start answering the questions in the chat while we're great.
Great. Are you able to see my deck?
Yes.
Great. Well, I come from a little bit different perspective on this, but the reason we're going to go to space here in this conversation is because spaceflight is actually a wonderful laboratory for accelerated aging. And so it's a great place to test your hypotheses. Are there ways in which we can alter the progression of aging and disease? But also there's probably a commercial pull for this technology in the future as well, because we're going to become a multi-planetary species. The move is already afoot, as you may be aware, with the new commercial spaceflight industry. That's a little bit off into the future, but I think looking at human physiology in space is actually quite instructive for this discussion. So a little bit about myself. I am a former NASA astronaut. I traveled into space on 5 space shuttle missions and did some really fun things, including a bunch of spacewalks, including some quite epic spacewalks. But I'm a physical explorer first and foremost. So I've been to some rather extraordinary places on our planet as well, to include the top of Mount Everest and the South Pole and deep beneath our oceans and other challenging places. And what I like to reflect on as a serial entrepreneur and inventor is that whenever we challenge ourselves as a species, they're invariably catalysts for disruptive innovation. It's very, very difficult to go into space, to sustain life, to extract meaningful science in that environment. But once you invent those technologies, they invariably translate back to meaningful roles here at home. So currently, I'm a co-founder and chief medical officer of a medical AI company based in the UK called Jabs MedTech, and I'm also involved in building a very disruptive electric vertical takeoff and land aircraft through a company called Onward Air. So let's go ahead and jump into the main storyline here, which is, why is spaceflight relevant to this conversation? Well, first and foremost, when human beings go into space, although it looks wonderful to be floating there and to gaze back upon our planet, it's actually quite taxing on the body, not only in the environment, but when we come back to a gravitational field, whether here on Earth or in some distant time to go reside on the moon or perhaps on Mars. So there are a number of things that we have to reckon with. First and foremost is radiation. And the further we get away from planet Earth, outside of the Earth's protective magnetic field, we're more susceptible to galactic cosmic radiation, to coronal mass ejections from our own sun. So that's a very important factor in terms of cataract development, cancer development later in life, and if the radiation event were substantial enough, it actually could cause acute radiation sickness and even death. Obviously, this is a closed environment, so there are isolation and confinement issues and cross-cultural issues with astronauts and cosmonauts from all over the world. Just the mere distance from Earth makes it difficult to essentially jump in an ambulance and get to definitive care in a trauma center or a major medical center. We have to deal with the medical situation there on the spot with the tools that we have on board, what we brought with us, or what we might be able to telementor or teleoperate there. Other aspects have to do with the lack of gravity. Reproduction and fetal development are likely to be a gravity-dependent phenomenon. If you think about the complexity of neural development, the folding that takes place in cell division as the spinal cord and the brain forms, will this function in the very same way if crews are having living out their lives in low Earth orbit, or perhaps in a partial gravity environment on the moon at 1/6 Earth gravity, or on Mars 1/3 gravity? Do we maintain our humanness, or is that a new species that would evolve over time? Motion sickness is a significant problem. Obviously, here on Earth, we have our inner ear that gives us our gravity vector. Little hair cells in our otoliths tell us which way is up and down, left and right, and forward and backwards. But in space, they're just floating kind of aimlessly. And so our eyes have to become our predominant sensor of vector. And that can be quite provocative to some people, especially the first couple of days in space, and then the first few days when you come back to a gravitational field. Also, in the absence of gravity, we experience fluid shifts. So here on Earth, we have blood pooling in our lower body as well as interstitial fluids. But in space, that also migrates into the central circulation. You may have seen astronauts in space who have a puffy face. Oftentimes, we experience runny noses and mild headaches. So that's a significant issue in the short term, but in longer term, we're seeing substantial changes in visual acuity. And this is called spaceflight-associated neuroocular syndrome, or SANS. And it's thought to be due to an elevation in intracranial pressure. And there aren't successful countermeasures for this yet. We also experience musculoskeletal deconditioning. Even just sitting here on this call, our bodies are doing work. Our muscles and bones are resisting the force of gravity, and there's maintenance of those structures. But in space, unless we exercise really deliberately, as you can see my friend Sunny Williams doing here, she's on a treadmill, actually running the Boston Marathon, believe it or not. She was in space during the Boston Marathon. She's a super runner, but we have to pull the body down onto the treadmill surface. Otherwise, you're just going to be flailing there if you move your legs in a running motion. Similarly, we can't take a weight stack with us into space. We have to use hydraulic systems and other solutions. There's also an issue with intervertebral disc disease. As we go into space, our spines straighten, and we can actually experience disc prolapses. I'm actually one of the first astronauts to have experienced this. I ruptured a lumbar disc in my spine and had to have that repaired, almost certainly as a result of my spaceflight exposures. In addition, it shouldn't come as a surprise, but our heart doesn't have to pump against a gravity gradient, a hydrostatic pressure column. So our heart muscle kind of goes on holiday, and it needs to recover a little bit after we get back home. There's certainly immune system dysfunction and delayed wound healing that we've seen. As a result of this unweighting of the spine and the muscles and bones, there's higher levels of calcium in the circulation in space. And so it's not uncommon to see kidney stones form, especially if people aren't staying hydrated up in space. and a couple of other things. Sleep disorders are not uncommon. You can't just look out the window from the International Space Station and tell whether or not it's time to go to bed or time to get up. We go around the Earth every 90 minutes, so we're seeing sunrises and sunsets 16 times a day. So the circadian rhythms of our body are substantially altered. And then other threats to this environment. We're living in a closed environment, so if there can be pockets of CO2 buildup that we call hypercapnia. And of course, if there were a fire or other toxic leak in a closed environment like this, it could be catastrophic for the spacecraft and its crew. And just to be complete, obviously, low Earth orbit is littered with decades of spacecraft and flexipane and washers and things that have come off boosters over the many years. And so it's sort of a shooting gallery up there with probably millions of different pieces of debris that are traveling very, very quickly in crossing orbits and pose a real threat to the spacecraft as well as their crews. So just talking a little bit about this accelerated aging. That may come as a surprise to some of you. You may have seen Interstellar. You may have thought, well, gosh, this relativistic time dilation that Einstein and others talk about, shouldn't people get younger when they travel in space? And that is, in fact, the case. If you were to travel closer to the speed of light, the closer you got to that magic milestone, time would really dilate relative to those living here on Earth. And you would come back younger than your twin, for example. And shown here in this photograph is actually a couple of my friends, Scott Kelly and Mark Kelly, identical twins. They're both Navy pilots and astronauts. Scott ended up flying the first very long duration flight aboard the International Space Station. I think it was 345 days, while his identical twin brother stayed on Earth. And it was a great test bed to look at the differences between space flight and terrestrial life, and it was fascinating to learn some of the issues that Scott faced that Mark Kelly did not. But just to give you a sense, Scott only came back 2/100ths of a second younger than his twin brother in space. So there really isn't an appreciable benefit to traveling at merely 17,500 miles an hour in Earth orbit. You need to go a lot faster to really see the benefits of that. But there are a number of areas where we can use spaceflight as a model for countermeasures, in fact, for the aging process. And of course, sarcopenia, or muscle wasting of aging, is very prominent in spaceflight, as is bone restructuring and decalcification, as occurs in elderly people, a process called osteoporosis. There's sleep disruptions, of course, that I mentioned. And then, of course, we're really concerned about the radiation environment, in particular when we get outside of Earth's emphasis, the Van Allen belts, and travel, say, to the moon or Mars, where there isn't an appreciable magnetic field. The potential damage to our DNA and our other, including mitochondrial DNA, is quite a high threat. And this could lead, obviously, to various forms of cancer as well as cataract formation. And then finally, I'll just point out that traveling in space actually does result in balance disorders. We're learning how to float and fly in microgravity, but when we come back to Earth, our inner ear has to re-equilibrate and become the primary sensor again. So it's actually quite a nice model for testing out countermeasures to the loss of balance as we age. So here are just a few examples of some countermeasures that are already in work for space travelers, as well as things that could potentially convert into real solutions here on Earth. But obviously, exercise is quite important to maintain our musculoskeletal system. And so this is a hydraulic system that my buddy Dan Burbank is using aboard the International Space Station. But there are other technologies, including lower body negative pressure. The suit that you see in the middle of this slide here actually pulls the astronaut down onto a boot plate, which is providing loading, which is good for the musculoskeletal system, but it's also applying a suction to the lower body and actually helping the body re-equilibrate in terms of fluid balance, and potentially helping reduce the risk of this vision issue that I mentioned earlier called SANS. Radiation is obviously a huge issue for our space travelers, but not insignificant for commercial pilots as well. And so there are pharmaceutical approaches to these things, as well as shielding and dietary and pharmacologic interventions that can be considered as well for the future. And finally, I'll just mention this for interest and completion, but I think that there are a number of other wonderful challenges for innovators when we start to consider deep space exploration. But how do we create the food that we're going to need on long duration planetary missions? We can't take everything that we would normally send up on a space shuttle or a Dragon capsule. We'll need to essentially develop new technologies in hydroponics and living off of the land for future crews. How can we recycle, make our closed environments more efficient so that we can convert our urine and other waste products into potable water and water that can be used for hydroponics, so that we don't have to take as much or extract as much from the local regolith. Oxygen generation and CO2 removal will be very, very important. Have to be able to recycle these scrubbing technologies to make it more efficient. One of the more interesting challenges will be medication and IV fluid support. We can't take a pharmacy that will last forever. So will we have the ability to create things that we need on Mars and on the lunar surface, and use 3D technology to generate the surgical tools that might be needed in an emergency but couldn't have been foreseen before we launched. And then another quite interesting challenge for innovators, we won't be able to take every specialty in medicine with us. We can't take a neurosurgeon and a gynecologic surgeon and an orthopedist on every mission. So how do we manage that? Do we use telemedicine, telemonitoring, teleoperation for these types of challenging cases that we might see in the future? But the real challenge, especially when we think about going to Mars, is that it could take 21 minutes for a one-way transmission and 21 minutes back to Earth. So we'll need to think about just-in-time training and other types of AI assisted technologies to support the initial settlers on Mars in particular. So coming to sort of the crux of this, I mentioned this briefly, but mitochondria are critical to aging as well as space travel. And as I've already mentioned, mitochondria are more susceptible to ionizing radiation than even our nuclear DNA. Not exactly sure why that's the case. There probably Ben may know the answer to that question better than I, and he's certainly one of the world's leading authorities in this area. But there are a number of very significant papers that have been written, including from the National Academies and a wonderful study reported in Cell that show that mitochondrial dysfunction is adversely impacted in spaceflight, and it could be tied to a number of different disease processes, to include diabetes and other metabolic energy balance issues in the body. So it's really important that we not only understand this, but potentially have mitigations to support not only future space travelers, but all of us here back on Earth. And I think Tom did a very nice job describing what we call a mitoclock. This is this 93-year-old mother and her damage score of 25. But, could we conduct these types of assessments on orbit and really get a sense of how quickly the space environment is affecting our mitochondrial health? Should we be banking our mitochondria and consider bank, obviously radiation protected and so on aboard distant spacecraft, such that if there were a major solar storm, that we could actually infuse mitochondria to support those crew members. One of our team members, this is Mike Snyder here on the right. Some of you probably know him. He's quite a remarkable scientist and serial entrepreneur, but he's deeply involved in metabolomic monitoring, among other things that he monitors. But to be able to create miniaturized technologies that we could take with us that would support not only future crews going great distances in our solar system, but think about how we might be able to deploy these types of systems even in rural parts of this country, let alone rural parts of the globe. So that, in a nutshell, is my view of space and mitochondria. But I'll pass it back to you, Ranjan, for any questions.
Let's go to our next speaker, Ben Albensi. As Scott said, yes, one of the world's experts in this area, in mitochondria and neurodegeneration. Ben, are you ready? Perfect.
Let me just share my screen. Can everyone hear me?
Yes. Off you go, Ben. Oh, you got to do full screen, yes. And then there might be some questions on the chat, Scott, on space. You have a quick look there.
Okay, great. So good morning, good afternoon, good evening, everyone. Depending where you are on the planet or above the planet, maybe some of Scott's friends are watching from the ISS. So I want to thank the R42 group for inviting me. I think I spoke to you folks about 3 years ago, so thank you for having me back again, and also to Matrix Bio for inviting me to speak today and to be a part of this. This is really exciting. And so, I'm going to talk a little bit about longevity in general, mitochondria more in particular. And I think what I'm doing really is tying together what Tom said and what Scott said to some degree. So, I've been studying, I'm a neuroscientist by training. I learned techniques like electrophysiology and medical imaging, got involved in memory and intelligence research about 30 years ago. And then about 18 years ago, I started studying Alzheimer's disease and how memory gets impaired. And then about 10 years ago, I jumped into the mitochondrial world and found it fascinating. And it seemed to really explain and help me understand beyond A beta and beyond tau, what might be happening to memory in Alzheimer's disease. So I got real excited about that. And of course, Tom contacted me, I think 3.5 years ago to do some studies with his team. So that's where I'm coming from. And, now that I'm in Florida for 2.5 years, I've been following NASA ever since John Glenn, but living here on the Space Coast, it makes me think more about what we do in my lab and how it actually can inform and potentially improve astronaut health. So I think there are really direct applications for space travel, and I think Scott alluded to some of those in his great talk. And so that's what I'm going to talk about today, especially this whole concept of accelerated aging. So here are my disclosures, and here's my agenda for the next 15 or 20 minutes. But like I said, in my lab, we study many of these space health related challenges, which I didn't originally think of as just space health. But there's so many things that we look at with regard to brain aging and brain changes and how space can accelerate aging. And of course, Alzheimer's disease is an age-related disease. So there's tremendous overlap. My focus is on some of those areas highlighted in yellow that you can see in this slide here. And so in particular, we work on memory loss, we look at brain connectivity changes, but we also study the immune system and the inflammatory processes. And both Tom and Scott talked about that, but we spent a lot of time in my lab focusing on proteins and other processes that contribute to inflammation in Alzheimer's. But it's really interesting because we see so much of this occurring in astronaut health too. And so a variety of factors affecting aging. And a lot of these changes are at the molecular and cellular level that we see in both Alzheimer's disease and in astronauts during space travel. So Alzheimer's disease is a classic example of an age-related disease. It's the most common type of dementia. And most of you out there in the audience already know that one of the hallmarks is memory impairments. But there are other things that happen, other changes, anatomical changes, behavioral changes that all kind of make an Alzheimer's disease patient who they are. And of course, back in 1906, Dr. Alois Alzheimer's was the first he did an autopsy and he found these abnormal deposits and really got the whole field of Alzheimer's disease research going. And what he saw in a neuropathological exam were these toxic, sticky amyloid plaques, and also these neurons that showed tangles. And this is the very first time these pathological hallmarks were really described in detail. So aging is the greatest risk factor for Alzheimer's disease, but we really have to remember that Alzheimer's disease is a multifactorial condition, so a whole bunch of contributing factors. So besides amyloid beta and tangles, we have other things that seem to really play a direct role, like inflammation, immune system changes, and of course, mitochondrial dysfunction. The other thing that's important to think about are sex-based differences in the occurrence of Alzheimer's, and we can't quite explain this yet by just longevity alone. In fact, what I'm trying to say is that women seem to, even though women seem to live longer than men, about 2/3 of all those with Alzheimer's in the US and Canada have Alzheimer's are women. So women are, at lower risk for Alzheimer's as compared to men before menopause, but right after menopause, their risk triples. So there's a huge difference, and many people believe this is due to the change in sex hormones. So, with testosterone, the male sex hormone, there's a gradual decline in this as we age. But in estrogen and other types of female sex hormones, there's a rapid decline in estrogen right after menopause. So I have a PhD student studying this right now, and so this is very much a focus of some of the work we do. And so, for example, A-beta deposits are found to be more striking in females, but we don't quite know why this is, and there's a lot of other sex differences too that I don't have time to talk about. But to this end, we've looked at sex differences in my lab in a variety of different mouse models. We have a clinical trial that just ended in Canada, and we're looking at sex differences there too. And so we found that there are many sex-based differences very early on in our transgenic mouse models in mitochondria. And we didn't expect this. We didn't expect this at all. And in fact, when we first started, this was very novel. And I think now studying sex differences has become very trendy. But anyway, sex differences also exist in space. So this is another interesting aspect that overlaps with what we do. And so, for example, male mice that are exposed to energetic helium ions show higher rates of neuroinflammation. And then studies have shown that female mice also have neurocognitive effects related to radiation. So with regard to female astronauts, women also show greater loss of plasma volume. Women astronauts have differences in heart rate and vascular resistance as compared to male astronauts. So there's a host of differences that we have to pay attention to. So as Scott alluded to and Tom talked about a bit, space travel also disrupts mitochondrial function. We see this in Alzheimer's disease. We see it in other age-related conditions as well. And so because of the studies, there's a lot of different mitochondrial markers that people are studying. Many of these are proteins, sometimes it's DNA. So we've been looking at these markers to ascertain mitochondrial health. And one of these markers that's been used in Alzheimer's disease, but also in space, are so-called cell-free circulating mitochondrial DNA, or CCF mitoDNA. So what they are is that they're short sections of mitochondrial DNA that are released by cells undergoing stress. So that's the important thing. So any sort of pathological event, the cell is stressed, they're going to release these short sections of mitochondrial DNA. So this can be used as an important marker. So studies have documented these changes in the CCF mitochondrial DNA in both astronauts and in Alzheimer's disease. Now, changes in markers of mitochondrial function can have important implications also for memory and memory encoding. And this is because synaptic plasticity and memory requires energy. And Tom talked a great deal about this. So, you know, the mitochondria are the biological batteries. And so this is a video one of my postdoctoral fellows created a couple of years ago, and the mitochondria have been labeled or tagged fluorescently with a dye, so you can visualize them as they move around. And so what we're going to see in just a few seconds, once I hit the start button, is that they're moving to sites of high energy demand. In other words, sites of synaptic plasticity. So let's see if this works. So, and the movement only occurs, though, if all the cellular machinery is in place. That is the mitochondrial proteins, and, you know, the mitochondrial DNA is doing what it's supposed to do, but we know that there's a lot of mutations in mitochondrial DNA, so that can affect function. And there are other proteins involved in mitochondrial biodynamics, too, that may or may not have been compromised. But overall, what I hope you saw in this short video is that mitochondria are moving to sites of high energy demand, and they do this a lot, much more than you think, and they're very rapid as they move around. So, you know, changes in cognition and memory, they're major hallmarks of Alzheimer's, but I really believe that it should be a huge subject of concern for long duration space travel. And I think Scott hinted at this. And in our lab and others, you know, many people use a variety of different methods for testing memory in human clinical trials or in our mouse models. And the top two left panels are a couple of the cognitive instruments that we use, the so-called MoCA test or the Montreal Cognitive Assessment Instrument. That's used, and it's found to be highly sensitive to MCI or mild cognitive impairment, which is the stage just before mild Alzheimer's. And we use it as a screening tool in our clinical trial. And so there's several domains that are tested using MoCA, visual, spatial, attention, verbal, and even math abilities. And then there's another test we've used more recently called CAN-TAB, and you can see that in the left middle diagram with those colored circles and squares. And that's it's a short test, 5-minute test that we use to measure attention, psychomotor coordination, reaction times, and so on. But these tests are used to measure cognition and memory. And there's a couple of others that are popular too. And then, of course, in animals, we use tests like the Morse water maze that we see in the top right corner. We use the Barnes platform. And then we use other things like electrophysiology and medical imaging techniques, PET, MRI scanning, to look at both functional changes and structural changes in the brain in both humans and animals. But so how are brain connections affected in space travel? Really, that's where I'm going to with this. And so in neuroscience, there's a concept, there's this thing that we refer to as the so-called default mode network, or DMN. And this helps us to understand memory, but also more specifically, brain connections. And so it's the DMN's integrity that appears to be crucial for memory and mental health. And it's interesting because in Alzheimer's disease, we see disruptions of this connectivity, this functional connectivity within different brain regions affecting the DMN. And this slide shows some differences in the so-called nodes between normal patients and patients with Alzheimer's disease. Well, interestingly enough, tests like this have also been done in astronauts, and some of the studies for space travel over 6 months, so a 6-month duration space flight, or I guess this would be on the ISS, studies have found that the DMN is disrupted, and some connections have thought to be disrupted in our astronauts as well. So why is this? And Scott talked a little bit about this. You know, levels of carbon dioxide are 10 times higher on the ISS. Radiation is a factor we don't fully understand, but 1 week on the ISS is almost like 1 year's exposure on Earth. So that's what, 40, 50 times more. And of course, microgravity introduces these fluid shifts in the brain and can affect ventricular volume, which then affects memory and cognition. And then also sleep deprivation. I think, Scott, it'd be interesting to hear you talk about this. But as I understand it, astronauts only sleep on average about 6 hours. And this is really not good. So, you know, one of the things that happens during sleep is not only do we consolidate memories, but we actually clear A beta from our brain. And so A beta is that sticky toxic protein we talked about. We're actually clearing that as we sleep. So sleep deprivation can accelerate amyloid deposition, which is going to accelerate memory loss, and impairment as well. And then finally, even long-term confinement, the distance from home, isolation is a concern. And there's some studies that show this affects neurogenesis, or in other words, the ability to grow new neurons, like in the olfactory bulb and in the hippocampus. So even neurogenesis seems to be affected because of one of the space stressors. So some of the countermeasures we can take, Scott talked a little bit about this. Well, of course, Tom is on to something really neat, and that's the bioreactor. And as part of the Mitrix team, we've been working on ways to grow and transfuse mitochondria for applications in my area, memory, but also cancer and sepsis, retinal degeneration that he talked a bit about. And so these are, up and coming ways that we might be able to control and reduce some of this pathology is to grow mitochondria, especially from our own bodies. The other things that I work on in my lab, diet and brain metabolism with the Alzheimer's Association. We're also studying repurposed drugs. And I found it interesting that Scott talked about bisphosphonates. I actually used to work on these when I worked at Pfizer in Michigan. And so, those drugs have been used for cancer. Well, there's a drug that we've been testing in my lab called nilotinib. It's actually a leukemia drug, and we find benefit in Alzheimer's disease. We find benefit in mitochondria. So, this might be one of those drugs that should be taken in a pharmacy toolkit for long duration space flight, so that you have drugs that might help to prevent and reduce the risk for both cancer and cognitive impairment. And so those are some of the things that we've been working on. So one of the things we attempted to do with Mitrix as a partner was to attempt mitochondrial transfusion in mice. And you can see that in one of our publications 2 years ago with one of my postdocs, Dr. Ad Limigadam, and then Tom was our sponsor. And we published this in the journal Molecular Neurobiology. And so there's also another recent paper by the Quebec group at the University of Laval, headed up by Eric Bolliard, where he's published a very interesting study with platelets in humans in the journal Transfusion. So, there's a lot to be done here. So I'm just going to talk a little bit about the data in our study that I just mentioned. So that's what I'm showing in this slide. So what we're doing is that we're using this method, a very common method called Western blotting, and it's used to measure protein quantity and quality. And so we're comparing the expression of protein subunits in the electron transport chain. You can see that cartoon on the right that shows the 5 main complexes. And what we found is that by using Western blotting, we were able to show that aging actually reduces the expression of these key protein subunits in the electron transport chain in complexes 1 through 5. As you compare 1-month-old mice to 12-month-old mice. So what am I saying? Well, in other words, when comparing these black bars of young mice to the gray bars of the older mice, you can see that protein expression of these complex subunits in the mitochondria go down in every case as we age, okay? So complexes 4 are very important because that's the site of oxygen consumption. Complex 5 is also important because that's the site of ATP generation. So in every complex I'm showing you there, it went down as a function of aging. Well, what we did next is that we took mitochondria from liver from these 1-month-old animals, and we injected these IV into 12-month-old animals. And you can see here that we found an enhancement in one of the mitochondrial complexes. That is complex 2 of the electron transport chain. And so what we found is that it actually enhanced that subunit that we detected in complex 2 with a couple different doses, and this was in the hippocampus. So it was a clear difference between in terms of the subunit expression in these older mice with an injection of mitochondria from younger mice. So this is very neat. And of course, this is just a starting point. This is in normal mice. The next step would be to test this in Alzheimer's mice or larger animals and eventually in humans.
So, Ben, a few minutes, and then we'll go.
Okay, let me wrap it up. One of the questions you might ask is if A beta and tangles, the hallmarks of Alzheimer's, are generated in astronauts during space travel. And the studies have been mixed. So in 3X mice, the type of transgenic mice we've used, there seems to be no change. But there's a study of 5 cosmonauts who spent about 169 days on the ISS, and they did show changes in neurological markers such as these glial markers, and also changes in tau and amyloid beta. This is reported in the journal JAMA Neurology. So just going to show this one last thing here. This is a timeline. A lot of physicians show this, Alzheimer's disease researchers. You can see a variety of different colored curves. Most people focus on the A beta and the tau. But what we find in our lab and what other people are finding is that these mitochondrial deficits occur much early in the process. So this is important, and it needs to be paid attention to both on Earth and Alzheimer's, and I think in astronauts. So to summarize, I think we can create brain resilience to memory impairment by enhancing immunity, lowering inflammation. We haven't talked about that, but I think it's important. These are important countermeasures for protecting brain function on Earth and in space. I think we also have to pay more attention to sex differences and sex hormones. And maybe we can achieve this with better diets, exercise, more sleep, specific drugs like nilotinib, bisphosphonates, and so on, and of course, mitochondrial transfusion. So I think I'll stop there. I just want to thank my staff in Canada that contributed to this work, especially with the transfusion project, and then my new staff in Florida and the team I'm building here. So thank you much for listening. Oh, and thanks for my funding sources, Alzheimer's Association and the NIH. So.